Okay, so the video that I have originally done cut off the first part of these notes, so these are going to be just the first little part of the DNA replication notes. So DNA, when DNA replicates, when you replicate something, that means you're copying it, and so this is just going to be basically the overall process of how DNA copies itself inside of a cell. So the question would be, when does it copy itself? And it's going to do this right before the cell divides, so that the two new cells each have their own complete copy of the genetic material. To do that, we've got step 1, step 2, and step 3, and unfortunately this process involves movement, and that's really hard to show on a still piece of paper, so just hang in there. We're going to get the paper part copied out or copied down, and then you guys will be watching some animations that hopefully will kind of pull it all together. Okay, so right in the middle of the DNA molecule in the nucleus of a cell, the first thing that happens is the base pairs split apart, and there's an enzyme that comes in and basically does this, and that enzyme is called helicase. So helicase unwinds the DNA. Kind of creative name. Ends in ASE, so you know it's an enzyme. And then helical, so it's pertaining to the fact it's unwinding the helix. And the reason why it's got to do that is there's another enzyme that's got to get in there and read the bases to make sure that it's adding and then add correct nucleotides to each side of the strand. So when it splits like that, it makes almost like a little bubble, okay? So you got a bubble here. And if you think about it, there's like two little V's at the edge of each side of the bubble, and so they call this the replication fork. And there's, again, one on each side of the bubble. With DNA polymerase, you have to know direction in terms of your 3' and 5'. So we talked about that with DNA structure during the last lecture. So 5' means that there's a phosphate group exposed on this side of the DNA. 3' means there's a sugar exposed, okay? You don't even need to know that, okay? But you do need to know that the left side, so to speak, of the DNA is slightly different structurally than the right side, okay? And enzymes always read DNA in a 3' to 5' direction. So 3' to 5' direction is how this little enzyme is going to read it. So if you'll notice, these 4 little bases were split apart here, and then they're still there. It's just what's happening is the bubble's getting bigger, okay, and it's going toward the ends of the DNA molecule. The enzyme that comes in and actually reads these base pairs is called DNA polymerase. So if you guys remember with our first unit, a polymer is something it's a long chain of something. And that's exactly what this little enzyme is going to do, is it's going to make a long new molecule of DNA. All right, and again, it reads in a 3' to 5' direction. And it's going to add 1 nucleotide at a time. All right, so essentially then what you've got is you've got multiple enzymes working on this DNA strand, all right? So one DNA polymerase is gonna work on this side, and then another one is gonna work on this side. So it's gonna read it in a 3' to 5' direction, and it's gonna add the correct base pair. So if there's an A, what is it gonna add? Good, it's gonna add a thymine. And it's not just adding a T or a thymine, but it's a whole nucleotide. So it's adding a phosphate, a sugar, and a base, okay? So the phosphates and sugars, we tend to just draw them like the sides of a ladder, if you remember that. So T with its little phosphate and sugar. When it reads C, which nucleotide is it going to put next to C? Okay, G. And again, it's going to add the phosphate and sugar. And then a G, it's going to add the phosphate and sugar. A C, a T, an A, a G, and an A. In that direction. On the other side, it's just going the opposite direction. So again, 3' to 5' is what it's going to read. So I forgot to mention that again as a review. If it's 3 here, it's 5 here. And if it's 5 here, it's 3 here. Okay, it's called anti-parallel. And that's why it reads in opposite directions. So it's going to add a T, a C, a T, an A, a G, a C, and an A. So you can just start to see 2 new molecules of DNA appearing, okay, in this bubble, this replication bubble. So if more time goes by, which is the third picture, what's happening is that the bubble's just getting bigger. All right, so these are the original ones we just filled in over here. So TCTAG, TCTAG, okay. And as it keeps unraveling toward the end of the DNA molecule, again, that DNA polymerase just keeps adding base pairs, all right? So let's go ahead and do that. Not just base pairs, sorry, nucleotides. It's adding in that direction. Okay, and up here, it's always gonna add 3 to 5. 3 to 5. The arrows don't really matter that much. Okay, so eventually what would happen is it would keep doing that until it reached the end of the DNA molecule, okay? All right, so one more thing to add here is that one thing you need to know for the AP exam is that eukaryote cells have multiple replication bubbles. They don't just have one like this, and the reason why is that they just have so much DNA. Like with us, we've got 6 billion base pairs that have to be replicated. So if you just had one bubble per chromosome, basically, which is what this represents, is like a single chromosome, it just would take way too long to replicate those chromosomes. Prokaryotes, however, they have a couple million base pairs, and so they can get away with just one replication bubble. Okay, so I have a picture of that. Grab a plain piece of paper. All right, so here is a eukaryotic chromosome, and we can tell, again, because we've got multiple replication bubbles. Okay, 1, 2, and 3. So they're forming along, you know, the DNA molecule. So the dark blue is the original molecule, and the light blue is are the new bases that are getting added. So new bases always get added to the inside. And then as time goes by, the bubbles just get bigger, okay? And then finally, they just fuse into each other. So you've always got original DNA on the outside, new DNA on the inside. Original DNA on the outside, new DNA on the inside. All right, so yours looked more like this, your notes. Okay, which we haven't talked about this one yet, but we will right now. Under the microscope, this is what the replication bubbles look like. So it's showing you here a chromosome that's got multiple bubbles. And again, it's just because eukaryotes have a lot of DNA. So it just makes it more efficient. Okay, on the next little part here. This is showing the results, okay, of what we were just doing. So the results, you get 2 new molecules of DNA. I think the genetic code might be a little different over here, but it's okay. We're gonna practice doing some base pairing. So we would say that the outer ones are the original DNA or the old DNA. Okay, so if you'll label that, that's basically where the next video picks up, okay? So go ahead and you can play the next YouTube video and continue the notes.